junctions. And there's two types within interior junctions we'll be looking at. The first is going to be a belt junction. And the second is going to be a spot junction. These are also known as anchoring junctions. So they will make links between adjacent cells, and they will also make links between cells and a basal layer or extracellular membrane. So they'll bind cells to each other, they'll bind cells onto their basal layers. The next ones we're going to look at are impermeable junctions. These have a different function, which we will explain. And the one we're going to look at today is going to be called a tight junction. And then lastly, also with a different function, is going to be communicating. And the communication junction we are looking at is going to be what's called a gap. So the first two we'll talk about are going to be our desmosomes, starting out with the belt desmosome. And for this diagram, we are going to pretend that cells are squares, because I'm not better at drawing than drawing this straight line. So if we have two adjacent cells, so two lipid bilayers, first one over here, second one over here, neighboring each other, bilayers are adjacent. Our first type of cellular junction is going to be what's called the belt junction. And it's going to be reliant on the filaments that we just heard about from Jose. These are going to be actin filaments. In a belt junction, these actin filaments, which I'm going to label in the red, are going to form a contractile band around the entire interior of the cell. So on the interior side of the cytoplasmic membrane, we're going to have bundles of actin microfilaments that can serve as structural support and provide a link between the rest of the cytoskeleton and the exterior membrane. In two adjacent cells, they're going to have contractile bands of these actin microfilaments right adjacent to each other. And connected between them is going to be fibrous protein. So they're going to link between where these belt desmosomes, these actin filaments, run on the interior of the cell. Between two cells, these are going to give an ability for them to maintain their structure. So this is our belt desmosome. We're going to erase this if everyone has it and draw the next one. Next one we will go over is our spot desmosome. I'm going to write desmosome, but I forgot. Oh. Now, a spot desmosome is going to have about the same function that a belt desmosome has. It's an anchor of the cytoskeletal structure. So once again, we have our two adjacent cells drawn in the simplest fashion that I can possibly draw them. And we have a protein plaque along the inner cytoplasmic side of the cells, adjacent to one another. In between those, we will have cross-linking proteins. I've already gone over transmembrane proteins. How they form into this layer, and then they can cross-link between each other. We have transmembrane proteins linking those together. And protruding out from these are going to be some of our intermediate filaments. So our larger filaments that we just heard about, protruding out into the cell. And these are also going to be able to provide structure for the cell. So the rest of the cytoskeletal components can use this as a linking anchor with which to work off of and carry out cellular functions. Spot desmosomes can also be attached to the basal layer or the extracellular matrix. You can have cells that are adjacent, anchored onto one another. We can have cells that are anchored onto this extracellular matrix, this basal layer. And they also have intermediate fibers coming out from them. And usually what this intermediate fiber is a keratin fiber. So both of these are adhering junctions. These are strictly for the maintenance of structural to hold cells together, proper orientation. Those are what our desmosome ones are going to carry out for the cell. Our third one's going to be a little bit different. One number before we go. The number of these desmosomes, these belt and spot desmosomes, are going to rely on the mechanical stress that those cells are undergoing. So if you have skin, heart, you will see a lot of spot desmosomes. They're under constant mechanical stress trying to maintain their shape against that. So they need a lot of anchoring points, these adhering junctions, to make sure they can stay in their proper orientation. That's understandable, right? So, all right, next is our impermeable junctions. We're going to do tight junctions. I'll draw the same cell again. So two lipid bilayers right next to each other. Place this just what a tight junction is a series between two adjacent cells of junction linking proteins. And it's slightly different than what you see in these guys. So these are for anchoring, for support of the cell. These are a band across between the two cells, made of protein, that is going to create an impermeable space. So if a solid or a fluid wants to move past here, really got no trouble. It can make its way through. But with this, it's going to prevent that movement between adjacent cells. So, we'll talk about the permeability of the cell membrane. This makes sure. We have a space between that's unable for solid and fluids to flow through. So it has to pass through the cell, since these are adjacent cells pushed together. If you have cells that are lined up and there are tight junctions between them, that is like a sheet that is going to be a selectively permeable barrier, preventing things from getting from one side of the cell to the other without being selective, by passing through either channels or through the cellular membrane. Now, one thing to note, these two adjacent cells do not share cytoplasmic fluid, and they do not share cell fluid. So they are connected here, but the cytoplasm within these two cells is not connected, just on the external. An interesting thing. This connection between these junction linking proteins is going to be calcium and magnesium dependent. Which actually makes sense. The external cellular space in between cells has a much higher concentration of calcium and magnesium than the internal cytoplasm. So these linkages will end up occurring between proteins once they are in that extracellular space, where there's high calcium and magnesium. It's kind of like a regulation. Proteins that go into there don't link until they're exposed to the exterior, and the calcium magnesium help carry out that linkage, forming this next junction. We have two junctions that were for structural support. We had one that was to maintain our barrier, and then we have one more. This is going to be a communicating junction, also a gap junction. And of the four that we talked about, gap junction is going to be the only one in which there's a connection between adjacent cells that is going to allow fluid passage. And what a communicating junction is going to be is a pore between adjacent cells, with which things can flow through. In our case of a gap junction, these are going to be pores formed from proteins, and this pore is going to be called a connexon. And connexon is formed from 6 connexin monomers. If you take a side view of it, it literally is going to look just like this. So 6 connexin monomers are going to form a connexon that will be embedded adjacently between two cells. And it'll form a pore in the center of it. That pore is 1.4 nanometers in diameter, and it'll allow things to pass through about 2 kilodaltons KD, not DK. 2 kilodaltons in size. We should go to talk a little bit about different proteins, like how big they are and things. But just to note, this is tiny. So most proteins are not going to be able to pass through this pore. Your nucleic acids are probably not going to pass through. So no genetic information is shared between cells. Most of the products of that genetic information are shared. What can be shared, however, are fluids. So our cytoplasm. Fluids, small solutes, and most importantly, ions. So ions can pass through this. Sodium, calcium, magnesium. So we say that two adjacent cells that are linked with a gap junction are electrically and chemically conjoined, or electrically and chemically linked. You will see a lot of these types of junctions in communication between large tissue types. So by them being able to pass ions between adjacent cells, they can communicate with one another faster than any other form of communication. So this hormones can't go this fast, neural impulses can't go this fast. This is a direct connection, the diffusion of ions from one to the other that can help carry a signal. In heart muscles, there are tons and tons of gap junctions. The reason for this is calcium is one of the signals to start a heart contraction. So when in one myocardial, a heart muscle cell, the contraction starts from a calcium influx, it will be able to flow to the next one. And that can also contract. So they can act as a whole unit communicating with one another due to these junctions. It's also a really energy efficient mechanism of communication. So it's relying simply on putting a pore in the membrane and allowing diffusion to do what it does. Let's see. And the last thing, cancer cells. One thing to note is that cancer cells typically do not have any gap junctions. So there's preventing the communication with neighboring cells adjacent to it. And this is thought to be one of the starting points of that cell going into its cancerous pathway and losing its regulation. So it loses communication with neighboring cells. So we had belt junction, or belt desmosomes, spot desmosomes, the ones used for anchoring and support. We have tight junctions to maintain our impermeable layer, or selectively permeable. We have gap junction, which is allowing our fastest, most efficient communication between adjacent cells. And that is really all I had to say. So if y'all have any questions, we'll take them. If not, y'all can go ahead and leave early.
What time is it?